we have today Dr. Annalicia Vaughan, and she's from the University of Queensland. Dr. Anna, she is a postdoc researcher with the University of Queensland Thoracic Research Centre, and she's completed her PhD in 2018. In her studies, she has been studying cellular toxicity response to lung epithelial cells to diesel and biodiesel emissions. And today she will talk to us about the cytotoxic, oxidative, and inflammatory potential of alternative diesel fuels on primary human bronchial epithelial cells. Thank you so much for being with us today, and we're looking forward to your talk.
Thank you for having me.
I'll stop sharing my screen, and
okay.
Here you go.
Excellent. Okay. All right, so thank you for the introduction. So I'm just going to talk to you today about some of the work that I did in my PhD. Now, I mostly looked at the effects of diesel emissions on human bronchial epithelial cells, but we wanted to look at how those responses would change with different alternative fuels, such as biodiesel or different fuel additives, because it changes the physical and chemical composition of diesel. And so just to get a better understanding about how diesel emissions interact with the airways. But also under the context of different diseases, so COPD or lung cancer, we wanted to understand the impact of these emissions on vulnerable airways. So that's the general gist of what my PhD was about. I'm going to share with you some of my data from that. And then just to talk about the team that I work with. So I work at the University of Queensland's Thoracic Research Centre. That's based at the Prince Charles Hospital, which is on the north side of Brisbane. As you can see, it's my team here. We have a lot of females in our team. We have one lone Y chromosome over here, and we actually have another research assistant that's recently joined our team as well. There we go. Yeah, so just a little bit of my background. I have a Bachelor in Science, majoring in Biomedical Science, and I got that in 2012, did my honours in 2013, and did my PhD about 2014 to 2018. And essentially that looked at the toxicity of diesel emissions on human bronchial epithelial cells. I was very lucky to be awarded the Prince Charles Hospital Foundation Research Fellowship. I was awarded that in 2017 and started my placement as a postdoc in 2018. And essentially that work is looking at, it's a little bit different, it's more like clinical study. I'm doing a randomized control trial, and we're essentially looking at dietary fibre as a therapeutic agent to reduce inflammation in COPD. But I'm still very passionate about looking at aerosol toxicity and the effects of pollution on the lungs, and especially with those with underlying lung disease, those with chronic lung disease such as COPD, asthma, and cancer as well. But I want to focus today on a patient population, those with chronic obstructive pulmonary disease, or COPD. So for those of you that are not aware, chronic obstructive pulmonary disease is the third leading cause of death worldwide. I think that's current as of this year, but it's predicted at least for 2030. And it's characterized by irreversible airflow obstruction. So these patients have a lot of trouble. They can breathe in reasonably okay, but exhaling breath is quite a struggle for them. So their airways are obstructed and the airflow in their lungs is limited. It's also characterized by chronic inflammation. It's one of the leading causes of the progression of disease, but also the airway remodeling, which changes creates the structural changes of the disease. And one of the causes of this chronic inflammation is the persistent exposure to noxious inhalants. So one of those might be cigarette smoke, but we also think that one of these inhalants that can lead to the damage that we see, or the chronic inflammation that we see, is airway pollution. It's just not as easily kind of monitored or tracked in the same way we can track someone's smoking history. So we do believe that one of the main contributors as well to the development of COPD is ambient air pollution. It's also a very complex disease. It requires multifactorial a stepwise approach to treatment. It's usually a team of people that help their patients manage their disease. There might be physiotherapists, exercise physiologists, or dietitians, as well as their GP and their respiratory physician. The major risk factors for COPD are largely age. It is an aging cohort usually that develops COPD. Another major risk factor is, again, that chronic exposure to noxious inhalants. So apart from cigarette smoke, there are different occupational exposures like dust and fumes. If you worked in maybe the construction industry or as a mechanic, that kind of thing. Another major one would be biomass smoke. In a lot of developing countries where they use wood fire smoke to cook or heat their home, and it's not well ventilated, this biomass smoke has been attributed to the risk of COPD. Another big determinant is genetics. We know that some people can smoke their whole lives and not have a chronic lung disease, but others do. And there are other factors as well, such as comorbidities. These patients often have a high risk of developing lung cancer if they have COPD, but they also have a lot of other chronic diseases, quite often so cardiovascular disease or type 2 diabetes. So those things, their comorbidities might be at risk for developing COPD as well. So just a rundown about diesel particulate matter. Diesel particulate matter is considered to be the more toxic element to diesel emissions. That's the current kind of belief, so to speak, but we do have evidence and we publish on the potential for the gas phase. So if you remove the particles, that can also be very toxic to cells as well. But essentially, diesel particulate matter, there's because the combustion of the diesel engine the fuel is not fully combusted, it produces these emissions that contain particles and liquids and gases. So the particles contain both organic content, but also there's metallic content as well. And bound on the particle surface, like I said, are organic content, and they're considered also the more biologically reactive element to the particles, in that as they cover the surface of the particle, they interact with the different cells and the different components of the lung tissue. And I just wanted to draw your attention to this graph here. This is, we're going to kind of keep coming back to this. It's just a graph showing the kind of deposition pattern, so to speak, in the airways based on the size of the particles. So particulate matter, when it's inhaled into the lungs, how far it kind of deposits in the airways is largely dependent on the size of the particles. The bigger they are, the more likely they're going to get captured earlier on in the mucus layer and get removed with mucociliary beading. But those finer particles can actually travel further and deeper into the airways and potentially cross that mucus layer into epithelial cells. And so when we see this curve here, we have different curves here. If you look at this one here, this solid line is a deposition of particles in the airways. And I'll just draw your attention here to the grey line with the black dots here. Is labelled with nucleation mode. And the reason why these are important is because, first of all, they have the highest deposition rate in the lungs, in nucleation mode. They're the smaller, so we're talking about 10 nanometers in size. So they're smaller than your nanoparticles. But also the important part here is that because they are the smallest, they actually have the highest kind of quantity proportionally of organic content. If you look down here with our nucleation mode particles and accumulation mode particles, those accumulation mode particles that are a little bit bigger, just bigger than the ultrafine particles, they do have that carbon core and they have an organic content lining on the surface. But if you look at nucleation mode particles, because they're more combusted, they actually don't have that much of a carbon core, and they're largely a liquid particle, but liquid with organic content. So, like I said, highly biologically reactive. So this is a very basic diagram about what happens in terms of a cell response to diesel particulate matter, but it just highlights the main kind of aspects that I've looked at in my research to get your basic cell responses from the different fuels that I've tested. So essentially, the diesel particulate matter, if it's of an ultrafine size, it can potentially cross this mucus layer into the epithelial cell with the organic compounds bound, and usually there's some exogenous ROS, reactive oxygen species, on that particle as well that get introduced into the cell. And that can trigger changes in gene expression as well as your pro-inflammatory response, which can trigger production of xenobiotic metabolism, but also the production of endogenous ROS, which can lead to oxidative stress. And then ultimately this can lead to cell death and tissue injury, as well as an array of other responses. But these are the key first response kind of things that occur when the airways are exposed to diesel emissions. So the aim of my research, in general, is to investigate the effects of diesel emissions. But we're looking at diesel emissions with varied physical and chemical compositions, so we can kind of interrogate and find out a little bit more about what's happening when the diesel emissions are exposed to the airways. Now we use largely primary human bronchial epithelial cells, and I'll talk to you a little bit more in a bit about where we get those from. But we've used air-liquid interface exposures because I think it's incredibly important that when we're testing the toxicity of aerosols, that we're doing it in a way that's as physiologically relevant as possible. So the main downstream responses that we're looking at are cell viability, inflammation, and oxidative stress. So it's hypothesized that diesel emissions will result in a loss of cell viability, increased inflammation, increased oxidative stress response. But we also think that the magnitude of these responses will be dependent on the physical and chemical composition of the emissions. I'm just going to give you, because with all of my experiments, I'm going to take you through the general methodology that I use is consistent. So I'm going to take you through it once, and then I'll just talk to you about the specific differences in study design that I use, depending on what fuel type we're using and things like that. So this is our general overview of the methods that we've used. So we have our primary human bronchial epithelial cells. Now these are established from resected specimens from patients that are getting lung cancer resected from their lungs. So when a patient is diagnosed with cancer and it can't be treated with chemotherapy or radiation, they'll get a portion of their lung removed. So they might get a lobe removed or a whole lung. And when that happens, if the tumour is kind of distal to the bronchus of the lung, we will actually collect a piece of a bronchial ring, and we'll treat that and remove the epithelial layer on the inside of the bronch ring, and we will isolate and grow that cell line up. So we have a bunch of about 150 in-house cell lines. Now all these cell lines are from those patients that have been diagnosed with lung cancer. We haven't taken it from the site of lung cancer, but it's good to know that these aren't considered healthy or normal. I class them as non-COPD. If I'm looking at COPD, I class them as non-COPD cell lines, but there is that kind of limitation. We have looked at commercial cell lines, which I'll talk about a little bit later in the talk as well. And we grow these cells like an air-liquid interface, so they're differentiated into the physiologically relevant cell layer. The diesel emissions are exposed through the Coltex radial flow system. We generate the diesel emissions in real time in the diesel engine lab at the International Laboratory for Air Quality and Health, which is at Queensland University of Technology. All of my exposures are for 30 minutes. And downstream from that, after the exposure, after an incubation period to generate supinate for downstream testing, we test for cell toxicity, cytotoxicity, cell death, cell proliferation, some inflammation markers, so the secretion of IL-8, IL-6, and TNF-alpha, and we also look at antioxidant production as well. And these are just a few papers on the side here that just kind of reiterate the importance of establishing your cell culture method based on what it is that you are trying to ask. So for us, we want to know how these cells respond to the chemical or physical composition of emissions. So it's really important that we maintain the integrity of those emissions, So air liquid interface is really important for that. So again, just to reiterate the importance of ALI culture, I still read a lot of papers that use submerged culture for their work. But the problems with that is that they form like a monolayer structure. It's not super stratified. All the cells have a basal cell phenotype, so they don't have that mucociliary phenotype. They often use immortalized cell lines, so they're a little bit more robust than your primary cell lines. And also they suspend their particulate matter in media, which can change the physical and chemical composition. So if you're wanting to know about the response to that, you're finding a response to that emission source that's actually not representative of what's in the ambient air. But with the air liquid interface culture, you have your pseudostratified structure. You have your basal cells as well as your ciliated cells and your mucous goblet cells. You use primary cell lines to establish those cell cultures. So you can use like healthy or normal, but you can also use your disease cell lines as well. And of course, the main benefit is you actually expose your cells to the emissions themselves. So that's ultimately the important part. Next, I want to talk to you a little bit about the kind of equipment set up to run the exposure, create the emissions, and also some of the equipment we use to analyze the characteristics of the diesel emissions. So you can see here that we have a diesel engine. Now the diesel engine generates the emissions in real time. The diesel fuel we use in terms of the actual conventional diesel fuel is literally just this fuel that we go down to the Caltex and buy. So it's the commercial stuff that we use here in Australia, and we just modify it based on, like, we just add the biodiesel that we want or the fuel additive that we want to test. The diesel emissions are then diluted 1 in 100, because obviously no one's breathing directly straight from an exhaust. So what we are actually exposed to is diluted. But even 1 in 100 is really just kind of standing next to, semi-trailer truck essentially. So it's still like pretty high exposure, but still relevant. We do have 2 points for gas analysis, and that's where we mainly want to focus on CO2 output, because we have it right after the emissions of diesel, the diesel emissions, and then also at the end of our exposure system or analysis system. And that's pretty much just to confirm the concentration of the diesel in terms of, like, do we truly have a 1 in 100 dilution? We check the CO2 concentration before and after dilution. We have 3 main instruments we use to look at the particle characteristics. Now, when we're looking at particle characteristics, we're looking at particle size, particle number, and the average particle mass as well. So we have the SMPS, Scanning Mobility Particle Sizer, the Dust Track Aerosol Monitor, the Vitality Tandem Differential Mobility Analyzer, BTDMS. We also have a HEPA filter that we can put before and after our exposure aerosol delivery system. So if we wanted to test for gas phase or for all of our controls, our negative controls, we had filtered laboratory air. So we'll put a HEPA filter in front of that exposure system and expose to laboratory air. And then, of course, we have our Caltex radial flow system, which is our aerosol delivery system. This is it here, a bit of a close-up. This is from the brochure. You can see that there's a upper chamber and a lower chamber. There's also 3 exposure wells here. They're set up for, your transwell inserts or something similar. They have 3 wells in which you can put PBS or media into those wells, and a heated system so you can regulate that exposure environment to 37 degrees Celsius. And of course, there's 3 wells. This is kind of an older model, so you can run in triplicate, but the newer models like the Caltex Compact has 6, so you can run your filter air control simultaneously with your aerosol exposure as well. That's really helpful. Yes, it's Caltex radial flow system. You can see here on this figure here that the aerosol comes in through the upper level and is divided into the 3 exposure wells. So just a little bit more about what I do after the exposure. So the cells are exposed for half an hour to either their diesel emissions or their alternative diesel emissions or their filter control. And then they're incubated. I put media on top of the cell layer, 250 microliters. Then they're incubated for 3 to 6 hours. I'll explain the 3 to 6 depending on the study design, at 37 degrees Celsius. And then we collect the apical media as supernate for stuff like ELISAs or analyzing cytokine secretion, or looking at cell death if we're looking at LDH assay. And then we look at cellular metabolism using the cell layer. So we run a WST1 assay, which gives us a readout of whether cells are metabolizing or, and then we take the cell layer, we extract the RNA, and then that's when we can do our gene expression work with that. We do collect the basal media, but we don't use it for anything at this point. So just the overall of how this webinar, the rest of this webinar is going to go. I'm going to talk about 2 main projects, and then I will actually talk about a little bit of extra work that we recently completed on e-cigarettes. The first project is looking at there are cellular responses to diesel or alternative diesel emissions that have substituted a portion of the diesel fuel with either a crude oil, so crude coconut oil, or substituted some of the diesel fuel with a coconut oil derived biodiesel. So biodiesel is pretty much where a vegetable oil or some kind of food oil, animal fat or something has been chemically altered, usually with some kind of alcohol like ethanol, to transfer some of the organic groups, and so it mixes better with the fuel. So it blends better and also improves the combustion efficiency of the engine as well. It's a very common process to create biofuels just because it's better for the engine, and it often reduces the emissions of the diesel fuel, which I'm going to show you is good, but also there are some negatives to that as well, because it does alter the chemical composition pretty strongly. And then the other project I want to talk to you about is actually a large cohort study. So when I was doing my PhD, I actually didn't see a lot of aerosol toxicity work that used a really a decent sized cohort of patient or participant cell lines. They would be kind of limited to maybe 3 or maybe 5 at max. But I thought, because I have this biobank of patient cell lines, it would be really useful and interesting to actually do a decent sized cohort. So I'm going to look at how these cells responded to the diesel emissions. And the reason why this is important is because when you take primary cell lines, especially from a patient cohort, there's a lot of variability in how the cells will respond. So a lot of that is dictated by potentially, that person's kind of history and what their lungs are like when we collect these cells. So I thought it'd be really interesting to see what would happen there. And then, like I said, we've got a little extra work at the end, just looking at e-cigarette toxicity. So the first study is looking at coconut oil derived biodiesel. And so the aim is pretty much to compare the biodiesel emission response to biodiesel emissions to conventional diesel emissions, and to see what the cell responses are like, and are they good, or are they better? So I essentially looked at 6 different fuel types. We have 100% diesel emissions, 20% biodiesel, 80% diesel, and then that goes up to 50%. And then when we get up to the 90% biodiesel, what we actually do is we don't actually have any conventional diesel emissions. We actually transfer that out for triacetin. And the reason why we do that is because it increases the oxygen content of the fuel and helps with combustion of the fuel in the engine. And so we did that again because the engine might struggle a little bit with 90% biodiesel. And so we did it with 96% biodiesel as well, and 4% triacetin. And then we also did do the 100% biodiesel. And like I mentioned, the biodiesel itself is transesterified coconut oil. And the negative control we have is the filtered laboratory air. So essentially it's the same. The diesel emissions and the biodiesel emissions are diluted by the laboratory air. And so, yeah, we're just taking out that factor of the emissions and seeing what the cells are like, how they respond to just essentially clean air. Okay, so some of our diesel emission characteristics here. We have our carbon dioxide. This is obviously the endpoint carbon dioxide. So, and nitric oxide, particulate mass and particulate number. I just want to point out, so we have like 0.79 in our diesel as our mass, which is quite high, but it's still relevant. I will point out that in 2016, the peak ambient concentration of PM2.5 was 0.14 milligrams per meters cubed. And some of these values are close to that value. So we're talking about levels that are comparable to those high pollution cities like Beijing and things like that. 0.79 is quite high, but like I said, we're only diluting 1 in 100, so that's the equivalent kind of standing next to the truck while it's running. Yeah. So I just want to give you draw your attention to these graphs here on the right hand side, and just kind of draw. I think I have the next slide. Yeah. Draw your attention back to this graph that we have here. So we have this peak here for nucleation mode, and then again the highest deposition rate was in the nucleation mode particles, and they're down at the 10 diameter of 10 nanometers. So back to this graph, you can see we have a peak here for our diesel emissions, and then if you also look at B50 here, which is our 50% biodiesel, 50% diesel, you can see a peak in the nucleation particles as well. We do have our accumulation mode particles. Most of our particles are accumulation mode, but I just wanted to draw your attention to the diesel and the B50 have their nucleation mode particles, a high deposition in the lungs. They're also largely organic contents, are highly biologically reactive. And we have our B50, and also in this bottom graph here, I've graphed the fuel types that have triacetin versus diesel and separate graphs, just as ease for kind of interpretation. Again, we have B90 with our nucleation mode particles, and then again, that's the same for diesel as well. We have a little bit of a peak for B100, but nothing major for nucleation. Yeah, so essentially when you graph the changes in the emission characteristics over increasing percentage of biodiesel or increasing percentage of triacetin, for biodiesel we have a reduction in the particle mass, so the emissions are reducing. We have fewer emissions from our diesel particulate matter. When you increase the biodiesel percentage, kind of variable changes, we have a decrease in particle mass but increase in particle number. That just kind of says to us that, as predicted, the increase in triacetin improves combustion because the particles are getting smaller and there's more of them. So that just tells us, yes, our combustion is getting more efficient. We do see some increases in gas, which kind of level off a little bit with the high percentages of biodiesel and triacetin. So this first slide is looking at our biodiesel blends with diesel. I just wanted to point out specifically, so with diesel we have a drop in metabolism, there are changes in cell death, but some kind of reduction in apoptosis. So biomarkers for apoptosis is gene expression of caspase 3 and BCL2, but I just want to draw your attention mainly to B50 where we saw the nucleation mode particles. There's a significant increase in cell death, both compared to diesel and filtered air. And then we do see a suppression of BCL2, which is an anti-apoptotic biomarker for B50 as well. And that fuel type was our strongest responder. When we look at the biodiesel blend with triacetin instead of diesel, so we've got our B90, we see an increase in metabolism, but we also see an increase in cell death compared to diesel and filtered air. And again, we see that decrease in BCL2. So that decrease in anti-apoptotic marker and increase in cell death. And then again, there are two our fuel types that produce those nucleation mode particles. when we look at inflammation and antioxidant production, you can see here there's an increase. When we look at B90, there's an increase in IL-8 secretion, and there's also also an increase in IL-6 secretion. It's not significant, but there's a trend there. And if you look at here's heme-oxygenase 1, which is an antioxidant protein. If you look at the expression, we're seeing an increase in expression for both the B90 and B50. So again, our nucleation mode particles, if you were to summarize that, with high nucleation levels in our emissions, we do see an increase in cell toxicity, an increase in inflammation, an increase in antioxidant response. But the more important part is that the use of this biodiesel, coconut oil-based biodiesel, is that these adverse effects were actually significantly stronger than our diesel emissions and our filtered air. And so even though we saw a reduction in emissions, we saw a reduction in particulate matter, we saw a worsening of these cellular responses. And so a lot of the emphasis on improving air quality has been to reduce emissions, but we don't actually do a lot of biological testing to see the biological toxicity is of these potentially alternative emissions that we're introducing into the environment. So I think this is important because we need to start looking at that as a standard for what we're going to introduce into the environment in the future. Are we introducing something that's safe for human health? Even though there's reduced emissions, it might be good for the planet. Okay, so the next aspect of this work is to look at the effects of crude oil substitution in diesel fuel. So this is just the pure oil. It hasn't been transesterified to become a biodiesel. I will point out, I put this little note at the top of my slide. This work was done with 16 HBAs. I know I emphasized the importance of primary cells, but we were given 2 weeks notice to prepare for this, which is not enough time to develop the air liquid interface culture with primary cell lines. But this work is still done at air liquid interface. We just use the immortalized cell line. And so they were cultured for a week just to make sure they were happy at air liquid interface, and then we exposed their cells. Yeah, so the aim of this work was essentially to compare the response of bronchial epithelial cells to diesel emissions that had a percentage substituted with crude oil, specifically coconut oil. Now, I just want to tell you a little bit of an anecdote about how often you can prepare. We spend a lot of time preparing for these exposures because once we're in there, we want to be efficient. We only have a limited amount of time because it is a shared space. Other teams use it, so we put a lot of effort into preparing for our exposures. One thing we didn't consider is what coconut oil is, and potentially the melting point of coconut oil, and the fact that we had actually, when we were given the opportunity to do this was actually in the middle of winter in Brisbane, which isn't cold, but it's not hot either. So a little bit of an anecdote, we ended up finding out that, I use my own artistic license here, after our first day of exposures, we found out that our coconut oil was solidifying because it was below 24 degrees Celsius, which is the melting point for coconut oil. So we actually had to do, luckily I work with engineers, so they had ideas, plenty of ideas, as we made up a little kind of cozy for our fuel drums, and then we had, as safe as we possibly could, we had some heaters that we could put in there and just maintain the room temperature above 24 degrees Celsius. Otherwise, our oil would solidify and separate from the fuel, which was actually causing a lot of problems with the engine because we were freaking out because our engine was missing, and we were just, we were very confused for a little while. Anyway, so the fuel types that we tested for this we kept our percentages pretty low because we are dealing with crude oil. So we have 100% diesel, 10% coconut oil, 15% coconut oil, and 20% coconut oil, and then of course our filtered air control. We have our particle size distribution curve here, and you can see with the 20% coconut oil, there is with the green dots here, we do have that peak. It's a little bit above the 10 nanometers, but still within that kind of nucleation mode. still within that nucleation mode. So I'll draw your attention there to the 20% coconut oil. We have our gas phase here, but I just want to show you that essentially our particle mass stay fairly consistent. There was a bit of a drop when we get to 20%. And then our particle number, again, stay fairly much the same. You can see that in the curves there. The curves are pretty much very similar. Okay, so when we look at cell viability, we see a reduction in cell metabolism, which is partially recovered with 20% coconut oil, but we do see an increase in cell death compared to filtered air control. It's not significant compared to diesel, but it is higher than diesel. So just want to draw your attention there. No significant changes for our apoptotic and anti-apoptotic biomarkers. For inflammation, we do see an increase in IL-8 and IL-6 secretion compared to filtered air, and IL-6 is also compared to diesel as well. So we have a higher response to inflammatory response compared to diesel. I will point out though that when you look at the lower percentage coconut oil, there is a reduction that's comparable to the filtered air and significantly lower than diesel emissions as well. On the left-hand side here, we have the expression of CYP1A1, which is a xenobiotic metabolism marker, and they pretty much respond to the organic compounds that enter the cell from the diesel emissions. And it's pretty much the cell's way of operating to produce proteins that will break down those foreign agents, so those organic content on the particles. But that process, the activity of those proteins to break down those foreign agents produce reactive oxygen species. So that's a source of endogenous ROS that might cause oxidative stress. So if we have a high load of particulate matter in the cells, we might get a lot more oxidative stress based on this pathway. And as you'd expect, we do see increases in the expression of this protein, because obviously we would have a degree of particular organic content the cells are trying to break down, and that's significantly high compared to the filtered air control. When we look at our antioxidant production, we have superoxide dismutase 1 and 2, and then a hemoxygenase 1. We do see increases in our antioxidant production, SOD1, but no real differences between the emission types. Some are slightly higher than diesel. It's not too alarming to see an increase in production of antioxidants, because we actually want the cells to produce more antioxidants so that they can balance the presence of ROS within the cell and don't undergo oxidative stress. But pretty much what we're showing here is they do have a response to ROS present in cells, so they can create that homeostasis there. But the main point is it's fairly comparable to diesel emissions itself. And the moral of this story, I guess, so far, is that when we're looking at alternative diesel emissions that might be effective in reducing the adverse response of diesel emissions to the airways, there were, at the lower percentages of crude oil, we did see responses that were significantly lower to diesel emissions and comparable to filtered air. And so if we're looking for an alternative diesel fuel, it may be more appropriate to substitute some of the fuel with crude oil itself rather than using biodiesel. But also keep it limited, because we did see with the 20% coconut oil substitution that increase in those negative responses when compared to diesel emissions. So that's essentially the moral of that story. So that's project 1. Oh, I threw this in there as a little bit of a joke, because sometimes people use coconut oil for everything. And so apparently we can also fix our bank account and our boyfriends with coconut oil. Anyway, so the next project is doing a large cohort study. So the aim was actually to look at potential kind of therapeutic agent to help vulnerable airways deal with the onslaught of air pollution exposure, because they already have chronic disease. We're focusing on patients with COPD, but they also might live in an urban environment, but they can't control the kind of air pollution that they get exposed to. So we're looking at a large cohort study, and and exposing them to just diesel emissions. We have no alternative diesel emissions we're exposing here. But after the exposure, they do get a 3-hour treatment or administration of an antioxidant to see if that alleviates that negative response to diesel emissions. So essentially we had our non-COPD cohort. We call it non-COPD, like I remember that these cell lines are established from lungs that do have lung cancer in them. They're not from a lung cancer source, but they're not considered healthy or normal either, so we call them non-COPD, and then we have our COPD cohort. So we had 11 cell lines in this group, in the non-COPD group, and 14 in the COPD group. And they were fairly consistent with their lung function and smoking history. Yeah, so their cells were exposed for 30 minutes, and they underwent the treatment with an acetylcysteine, which is the antioxidant we used, for 3 hours at a concentration of 5 millimolar. This is our particle size distribution curve for the diesel emissions, fairly standard, nothing too crazy here. You will notice that there's no nucleation of the particles. They're largely accumulation mode, which is fairly normal. A particle mass is 0.39. If you're wondering about the variability in particle mass between our diesel, the likely explanation for that would just be that with any diesel engine, the combustion efficiency will change based on how often, like at what time point the engine is used, from when it was last serviced or maintained or things like that. If it has fresh lubricating oil, combustion will be better. So we'll have likely a smaller particle mass, things like that. Okay, when we look at cell death, there's no significant changes here. There are some increases as we'd expect for diesel emissions. I do show the non-COPD and the COPD groups along with we do collate them as well, just out of interest. So that's what those graphs are showing, but no significant changes in cell death for this one. When we look at inflammation, we do see significant increases in inflammation. It's not really changed by the presence of an antioxidant, but there is a trend for a reduction. So it's potential that antioxidants might help with the inflammatory response. Maybe we just need a different antioxidant for that. And when we look at expression with our non-COPD, we do see an increase in expression that's actually reduced with the NAC. And with COPD, we actually saw no changes in expression for our cytokine there. Now, when we look at our antioxidant production, with our non-COPD, we see an increase in expression, significant increase, which is attenuated fully with the NAC, as you'd expect, because that need for the production of antioxidants isn't there, because we have introduced an exogenous antioxidant. But I will show you that with diesel, especially with superoxide dismutase, we do see a significant reduction, not reduction, but suppression of the production in response to diesel emissions. And so what that kind of started to indicate to us is that potentially COPD patients have a deficiency in producing that antioxidant response that we need these cells to produce to protect them from exogenous and endogenous RLS that's produced in response to diesel emissions. And so COPD cells might actually be vulnerable to diesel emission exposure because of an inability to produce antioxidants when they're needed. The potential theory there. I also ran some nanostring gene expression panels on these cell lines because I wanted to get a little bit, kind of like enter a bit of a discovery mode and get a little bit more information about what could be happening in these cells. So I ran the intracellular signaling panel and the immunology panel. So a little under 800 genes were assessed with this analysis. But one of the most interesting things I found was an increase in expression for interferon signaling proteins. So that's in response to viruses and things like that. So immune response to viruses, but also a really strong increase in expression from these NCH class 2 proteins, or genes, sorry. And you can see how strong that signal is. And so when you combine an MHC class 2 signal with an interferon signaling, it kind of tells us that maybe these epithelial cells, they might be primed for an adaptive immune response. And when you're dealing with a inhaled agent that is kind of inert in that it's not a bacteria or virus that can kind of replicate or infect, it's just kind of native in the fact that it just needs to be removed but can cause inflammation. The most important kind of immune response we need there is the innate immune response and the pro-inflammatory. And so these cells might actually be kind of challenged if they're actually primed to respond with an adaptive immune response, which is not what's needed in this case. So that's another avenue in terms of, you know, how vulnerable airways might respond to diesel emissions and make them more vulnerable. That was an idea that we kind of delved into a little bit as well. So the next study I want to talk to you just, you know, quickly about, just show you some of the recent work that we've done, which is with e-cigarettes. It was done by a current PhD student, Hannah O'Farrell, and we've got a manuscript in preparation. But essentially, we want to look at this dual device, which is really common. It's available in the US, and we want to look at the cytotoxic, potential cytotoxic, and inflammatory and DNA damage that could be associated there. So overall protocol, e-cigarettes are, it's kind of a debate going on at the moment to see if e-cigarettes are appropriate smoking cessation tool, because some people can't quit smoking, but for the, you know, we really want to get them off cigarettes if we can. And if e-cigarettes are not as bad as cigarettes, is it potential that we can kind of transfer their habit to e-cigarettes in order to kind of alleviate some of those adverse effects? But we don't know how bad e-cigarettes are. So this is essentially the work that we are undertaking at the moment. These dual devices, though, they're kind of USB in shape, and they're really high content of nicotine based on their nicotine salt technology. In one cartridge of a dual device is one packet of cigarettes, and in a half an hour exposure for our e-cigarette model, we used about a third of a cartridge. So a third of a pack of cigarettes in half an hour. Imagine someone sitting there smoking or vaping for half an hour. Anyway, so essentially same model. We have our in-house epithelial cells. We do use commercial cell lines in this exposure as well. But I'm going to make this really quick. And essentially, we just want to look at those basic cell responses to e-cigarettes versus cigarette smoke and see if they're good or bad. So we have our incubated cell lines. This is just the baseline cell. We have our filtered laboratory air, a positive control, which is cigarette smoke. And we use the World Health Organization's standard operating procedure for doing cigarette smoke exposure. And then we used we tested 2 e-cigarettes with the dual device. So one was tobacco flavour and the other one was menthol flavoured. This is just a diagram of our kind of system we use, but essentially it's just the puffing system. And so it was a 5-second puff duration. It's actually 30-second rest interval. Let's use that tab there. This is a little bit of a model. Just see if we can play this video. Yeah. So essentially, that's the HEPA filter. It gets filtered through this tube down to the Caltech radial flow system, where we can activate the computer here, which is this is our concentration of particles, just telling us that that's the model that we're using. And then yes, there's particles flowing through the system. Yeah, just a cute little video. Again, same system protocol we used before, primary HVACs, but this time we used the TER measurements, and we're actually looking at DNA damage as well. So we used commercial cell lines NHBE and COPD. We did have a lot of trouble with these cell lines. I'm not a fan of these. If you have any questions about them, hit me up. I won't go into too much detail. We had 4 of our lung cancer COPD cell lines as well. And the main story I want to tell here is, so we're looking at cell layer permeability. So it's based on our resistance readings here. The higher the resistance, the more tight the cell lines, and so kind of like more happy or stable the cell line is. So if we look at our pre and post exposure for the different cell types, which are the different colors of the bars of the different cell types, and if we look at cigarette smoke and our two dual devices, you can see there's a significant reduction in the permeability or the resistance in the cell layer, which indicates an increase in permeability for both the cigarette smoke and the two dual e-cigarette exposures. When we look at cell death, we can see here that when we look at cigarette smoke, we have our, I'm just going to focus on the COPD and lung cancer cell lines. This is a light grey bar here. You can see that there's cigarette smoke, there's an increase in cell death for the tobacco and a slight decrease. It's not significant, but there is a trend for the increase in cell death between cigarette smoke and tobacco e-cigarette vapor. When we look at DNA damage, again, I'll just focus on the COPD and lung cancer cell lines. We do see an increase in DNA damage compared to the tobacco e-cigarette, compared to the actual cigarette itself. And we also do a comparable DNA damage for the menthol. So that's telling us that these e-cigarettes are causing just as much or more DNA damage than the cigarette smoke itself. And then when we look at inflammation, again, we'll focus on the lung cancer and COPD cell lines. We do see comparable levels of inflammation, but they're comparable to our controls as well, so that's fine. When we look at IL-8, however, IL-6, sorry, we do see increases, a decrease for the tobacco, but an increase in the menthol in terms of inflammation. So there is some early evidence to suggest that e-cigarettes, if they're not comparable to cigarette exposure, cigarette smoke, they are worse than cigarette smoke themselves. Again, this is TNF alpha, very similar results there. So essentially, when we want to know if e-cigarette vapor are better or worse than cigarettes or the same, essentially we do see a decrease in resistance, so an increase in cell permeability, increases in cell death, inflammation, and DNA damage. So this early evidence is suggesting that they really shouldn't be used as a smoking cessation tool, because they could potentially lead to those tissue remodeling that could lead to chronic lung disease. And so in summary, for this work so far, if you see an increase in emissions, it doesn't necessarily mean there's a decrease in the adverse effects of this emission to airway epithelial cells. And so again, it's really important that we do look into those biological toxicity effects a bit more. Crude oil substitution may be a better alternative to biodiesel. And then of course, e-cigarette vapor may not be safer than cigarette smoke as a smoking cessation tool. All right, so that's my talk. I just wanted to thank the International Laboratory for Air Quality and Health and the engineering team there. I couldn't do any of this work without them. They tell me everything about aerosol physics and chemistry and engineering. So, and of course our patients that donate, part of themselves to our research and allow us to do what we do. Yeah, any questions?
Thank you, Dr. Annalisa. Thanks so much for the very informative and interesting talk. And we'll leave the platform now for our audience if you have any questions for Dr. Annalisa at the end. You can type your questions at the chat, or you can just unmute yourself and ask the question if you have any. We have one question here from Rachel. So Rachel, she's saying, fantastic work and presentation, thank you. Are you going to examine other forms of biodiesel?
I think for me at this stage, I'm not looking at biodiesel. I'm not looking at continuing my biodiesel work. I am very interested though in biomass smoke. I'm hoping to get a grant to look a bit further into biomass smoke, especially with the recent bushfires and things like that. I think there's a lot that we need to learn about the way the biomass smoke interacts in the airways, and whether or not we're doing enough to protect the airways of those high-risk individuals, like our emergency workers and things like that too. But if the team at the International Laboratory for Air Quality and Health, if they need to do cell toxicity work on any of their alternative emissions, I'll happily do any of those. But yeah, no, nothing planned just yet.
We have another question from Christian. She's saying, hi, Alicia, that was a great presentation. Thanks. Do you use the duster track to look at particle size and mass, or do you also collect PM with it?
We just use it as a monitoring tool. It's something we use in real time, just so we can, in real time, track the particle mass. Because if we see sudden increases or decreases, that'll give us an indication of something is happening with the engine or with the fuel that we need to address. But it also tracks the particle mass, so we can get the average particle mass for our data as well. But we don't collect with the dust track. No.
Another question from Brendan. He's saying, hi, really interesting stuff. Have you worked with fuel standards people in the Commonwealth Department of Industry and diesel engine suppliers to determine if the blends you have tested can ever be used in Australia?
I personally haven't, but I do believe that our collaborative group, the International Laboratory for Air Quality and Health, they're part of like an initiative by the World Health Organization. So they do a lot of commercial and industrial work with different entities to test different fuel types. So they might have worked, with that group, but I haven't personally, no.
Another question from Rachel. She's asking you, do you have any thoughts on why there are different responses to different concentrations of biodiesel blends with diesel?
I think that's entirely just because the change in the emissions. Like we did see some subtle changes in gas concentrations, nothing major, but the biggest thing that we saw was essentially the change in the particles. I think there could be a change in the deposition of the particles in our system. So if they get smaller, if they do become smaller and higher in number, they might deposit differently, either more effectively or less effectively. I think that would be the biggest answer, the most accurate answer is that the particles change. Their particles change in size. And when you change the particle size, you actually change the ratio of organic content on that particle as well. So that's one thing we didn't get a chance to look at with aerosol mass spectrometry, was actually the chemical breakdown of the organic on the part of the particles themselves. We do the chemical breakdown of gas, so to speak, but not so much the particles. So we don't know how the organic content changed with the percentage of the biodiesel, the crude coconut oil. So that would be my hypothesis, would be that, yeah, those organic compounds might have changed.
We have the last question from Jeff. He's asking you, have you had examples of people interpreting your results as not requiring decrease in PM emissions?
So I don't quite understand the question, sorry. Can you rephrase it? Well, Jeffrey, would you mind rephrase it?
Jeff.
Examples of people interpreting the results is not requiring.
Jeff, do you like to ask Annalicia the question yourself?
Okay, yes. Look, I may have missed the content, the detail of what you were saying, but I thought you said there was an inverse association between PM emissions and one of your markers.
Yeah, so our hypothesis essentially was that we would see a reduction in emissions, or that the magnitude of our response would be dependent on the physical or chemical composition of the emissions, right? And so under the current kind of belief, so to speak, or hypothesis that the particulate matter are the most kind of toxic component of diesel emissions, we had assumed that when you reduce the diesel emissions, you would see a reduction in the adverse effects that these emissions produce. And so that's a large assumption in like this industry, that if you reduce emissions, they're gonna be better for the environment, but also better for health as well. But what we actually saw was that even with a reduction in the particle emissions, we did see significant increases in cell death and inflammation and oxidative stress. I'm not sure if there was just like that kind of unexpected finding that was that contradicts the current belief or assumptions about the role that particulate matter play in these adverse effects. Does that answer your question?
It's sort of interesting because there's quite a wealth of evidence suggesting that particulates have substantial health impacts.
Yeah. It's not that I'm not saying that they don't, but it's just that I think that there's something else there. I think it's largely, I wouldn't be surprised if we do a little bit more work in gas phase and the role that gas phase plays. I did do some work looking at the oxidative potential of gas phase, so that we do have some evidence to show that the gas phase, and what I mean by gas phase is when you use a filter to remove the particulate matter and expose the cells to just the gas components of the emissions. There are some toxic effects on both like cell death and also inflammation from those gas phase alone. And when you reduce the particulate matter of the emissions, you actually allow for kind of the gas particles themselves are dispersed differently, and there's more gas that's like kind of provided or administered to the cells. And so, and it could be because it is a gas phase, they could have more kind of, the magnitude of their response could be higher because there's nothing in terms of a physical barrier, so to speak, that, well, to be honest, we just don't know, to be honest, if this gas phase is more or less toxic or how it interacts. I think we have more of an understanding about the particles, but I think what this data is suggesting that there's more to the story than just the particulate matter that we need to look at. So maybe reducing the particulate matter is a good thing, but we also need to understand what else is happening in the emissions and how they interact with the airways. Is that clear? So I'm not saying that particulate matter that they're not the toxic agent, but I think there's something else in the emissions that we've missed potentially, and we need to investigate further.
I suppose as a health person, I sort of get concerned about mixed messages around these sorts of things.
Yeah. Okay.
I just have a comment here saying I would add that PM emissions do not necessarily reflect the exposure. It can all be about the number of small particles that do not contribute to the mass much. Last question, we have another question from Christian, and she's asking you, do you run any studies using PM that you have collected on filters? And if so, what have you used to collect the PM?
I did. So we haven't done any PM work that was collected on filters. If we were to collect particulate matter and have them resuspended in media, we would use kind of an impinger system where we kind of, the emissions are impinged through the liquid and kind of collected into the liquid directly, rather than collecting them on a filter and then removing them from the filter and suspending. It's not the most efficient way to collect particulate matter, which is why we never kind of used it, because it wasn't a very high dose. But for my PhD, I really wanted to focus on, the air-liquid interface and really do some aerosol delivery and toxicity work, because there is a large body of work in terms of diesel emissions. And a lot of the research that comes out, a lot of it do use still the conventional cell exposure method. And so I just really wanted to focus on this air-liquid interface and see what data we got and see how it compares to the research that's out there.
Thank you so much, Annalicia, for your time today and your informative presentation, and thank you for all our audience. And we're looking forward to have you again in CAR. Thank you so much.
Thank you.
Thank you. Bye.
Bye.
Thank you.